Welcome students. Let's start our today's topic from the chapter cell, that is prokaryotic cell and its components. Students, prokaryotic cells are represented mainly by bacteria, blue-green algae and mycoplasma or EPLO. As per our previous discussion, the bacteria are of various types on the basis of shapes, such as they may be bacillus if they are rod-like, they may be coccus if they are spherical, they may be vibrio if they are comma-shaped, and they may be spirillum if they are spiral-shaped. Students, main component of prokaryotic or bacterial cell are: number one, cell envelopes; number two, cytoplasm; number three, its DNA, genomic DNA. Beside this, it has extra chromosomal DNA, that is plasmid DNA, cell organelle, inclusions, flagella, pili, and fimbriae. Let's observe the diagram, students. If we see the diagram of bacterial cell, it has many types of wall layers that are collectively called cell envelopes. It has, student, cell wall, it has cell membrane, it has glycocalyx. These all are collectively called cell envelopes. Beside this, student, it has cytoplasmic semifluid matrix. It has, student, genomic DNA, that is the main chromosomal DNA. It has plasmid DNA, that is extra chromosomal DNA. And cell organelle, if we talked about, it has only and only ribosomes, that is non-membranous cell organelle. Beside this, student, it has some bodies which are used for storing purpose, that are called inclusions. It has, student, flagella for the locomotion purpose, and pili and fimbriae, which are mainly used for adhering purpose. Let's start to discuss the first component of prokaryotic cell, that is cell envelopes. Cell envelopes mainly include three wall layers: the outermost glycolex, the middle is cell wall, and the innermost plasma membrane or cell membrane. Let's start to discuss the outermost layer, that is glycocalyx. It is simply called sugar coat, and it differs in its composition and thickness among different bacteria. Student, it could be loose sheath called the slime layer, and it may be thick and tough called the capsule. It is composed of polysaccharides, that is carbohydrates or proteins, and its main function is protection, and it also helps the cell to adhere to the different types of surfaces. Now let's start to discuss cell wall. Cell wall lies outside plasma membrane or cell membrane, and its main function is to protect the cell from bursting when it is placed in hypertonic solution. Sorry, not in hypertonic, but hypotonic solution, or in less concentrated solution, because we know that once any cell is placed in hypotonic solution, what will happen? Water will start to enter into the cell. So cell wall is very tough and rigid, so it protects the cell from bursting in that condition. Student, cell wall is of various thickness. In gram positive bacteria, it is of 20 to 80 nanometer in thickness, and it is single layered and smooth. Whereas in gram-negative bacteria, students, it is 8 to 12 nanometer thick. So what does it mean? That in case of gram-negative bacteria, the cell wall is thin. What are these gram-negative and gram-positive bacteria? These we will discuss in our next lecture. Let's start to discuss, students, now the third component of the cell envelope, that is cell membrane or plasma membrane. Student, as per of our previous class knowledge, cell membrane is selectively permeable or semi-permeable, and because it allows some materials to move in and out of the cell, and its structure is similar in eukaryotes as well. Students, there are some membranous extensions of cell membrane, and out of that membranous extensions, the mesosome is the most important. What does it mean? That sometimes cell membrane is extended toward inner side of the cell to form some specialized structure. So what is mesosome? Mesosome is a special membranous structure which is formed by extensions of the plasma membrane into the cell in the form of vesicles, tubules, and lamellae. So mesosomes are special membranous structures formed by the extensions of the cell membrane. The main functions of mesosome are: number one, these extensions help in synthesis of cell wall and replication of DNA. Replication of DNA means DNA copying. Number two function, mesosome also help in equal distribution of chromosomes into the daughter cells during the cell division. Third, it provide surface area for various type of enzymatic activities. And the last function is it helps in secretion processes, as well as the very important function that it helps in bacterial respiration. Its role is in respiration same as mitochondria plays its role in case of eukaryotic cells. So what are the important functions? Number one, it helps in synthesis of cell wall and replication of DNA. Second, it helps in equal distribution of chromosomes. Third, it provides surface for enzymatic activities. And fourth, it plays a role in secretion processes and bacterial respiration. Students, in some prokaryotes, especially cyanobacteria, there is another membranous extension that are called chromatophores. And these chromatophores, student, have a same role as chlorophyll plays in case of plants. What does it means? That chromatophores help in photosynthesis. So student, here we have finished all the topics related to cell envelopes. We have covered in cell envelopes. We have discussed about cell wall. We have discussed about cell membrane. We have discussed about the outermost structure, that is glycocalyx, and we have also discussed some membranous modifications in terms of mesosome and chromatophores. So here we have finished our first component of prokaryotic cell, that is cell envelopes. Now the second component is, that is cytoplasm, and cytoplasm is nothing, the fluid matrix that is filling inside the cell. With this, students, we are going to end our today's lecture. In this lecture, we have discussed first two components of prokaryotic cell, that is cell envelopes and cytoplasm. The most important one was cell envelopes, in which we have seen that cell envelopes is containing three-layered structure. The outermost is glycocalyx, the middle one is cell wall, and the innermost is cell membrane. The rest of the components we will discuss in our next lecture. Till then, take care. Thanks.